We see here the brain, the sheep brain. Here we are looking at a superior view of the sheep brain. In the superior view, we can see the cerebrum, which is our large section here, and we can see our cerebellum to the posterior, and we can see projecting out the posterior the spinal cord. Let's focus on the cerebrum. You can see with the cerebrum that the cerebrum has all of these hills and valleys. The upfolds, the hills, gyrus. The valleys in between, where you may see blood vessels going in here, are the sulcus. The sheep brain does have a central sulcus, at least this one has a pretty good one right here. The cerebrum is divided into two hemispheres. You have the right hemisphere here, and you have the left hemisphere here. These two hemispheres are divided by the longitudinal fissure. Between the cerebrum and the cerebellum, you will have the transverse fissure. Each hemisphere also has lobes. So to the anterior, you have the frontal lobe. Here we have the parietal lobe. Here we have the occipital lobes to the posterior. And to the lateral side, we have the temporal lobe. Let's turn our brain over and look at the inferior view of the brain. On the inferior view of the brain, you can see that when they took this brain out of the head of the sheep, they removed most of the meninges, the protective covering that we would find around the brain, but they left the meninges intact here on this inferior surface. They did this so that they could protect and retain many of the cranial nerves that you'll find underneath here. So we're going to eventually here remove this very, very carefully so that we can also retain many of the cranial nerves underneath here. But before we do that, let's look at the meninges. The outer covering here, which is very tough and durable, is the dura mater. If we take this and look at the inside of it, you will see that the inside is a little bit more smooth and shiny. This smooth, shiny inside is the arachnoid. The pia mater is attached tightly to the brain itself. And you cannot remove this pia mater without actually damaging the brain tissue. Okay. It will go down into the sulcus and over the top of the gyrus. A couple other structures before we remove these meninges. The first one is this structure that you see right here, kind of in the center of our brain right here. This structure is the pituitary gland. So again, the pituitary gland would be just inferior to the brain. When we remove these meninges, the pituitary gland will be removed with them. So we will remove the pituitary gland as we remove the meninges. On either side here, you may see part of cranial nerve number 5, our trigeminal nerve. Okay, remember our trigeminal nerve is very large and it branches out. So you may see part of that along the sides. Most of this we will remove, but we are going to hopefully retain a little bit of the stump as it enters and exits into the brain. So hopefully we'll keep that. So how are we going to remove these meninges? Well, first of all, we're going to do it very carefully. And to do this, we are going to sort of peel the meninges from posterior to anterior. And we're going to use our scissors to cut any attachments that you will find near the meninges and not near the brain. Most of the attachments are going to be cranial nerves, and you want to keep those with the brain and not the meninges. You want to take special care as you go to this area around the transverse fissure. And the reason for this is right here is where the trigeminal nerve will come off, but in the transverse fissure you will find your 4th cranial nerve, your trochlear nerve, and your trochlear nerve is very small and very tiny and very delicate. And on this side, it's already been broken off. I don't have one on this side. Let's see if I have one on the other side. And I do. It is right here. Can we see that? This teeny tiny little thing that I'm sort of pulling on right here. I don't want to pull on it before I cut it because it's super tiny. So again, depending upon who removed this and how good of a job they did, you may or may not have any cranial nerve number 4. Okay, because they get broken very easily. So I'm going to go in here and cut this one very close to the meninges. And there I got it. I'm going to pull towards the anterior, and again, you guys will do this also. Just a little at a time. Any attachments you want to cut near the meninges. Until you removed it, like I've done here. So as you removed the meninges, remember I told you that you were going to be removing the pituitary gland with it. So you have removed the pituitary gland from its attachment to the brain. So it's going to stay at this point with the meninges. So let's look at this brain. We're going to start with the cranial nerves. And you're responsible for knowing the first 5 cranial nerves, because usually that's all that's intact on these brains. The first cranial nerve is this one right here, and this is our olfactory bulb, cranial nerve 1. Now, many times when they remove the olfactory nerve, again, from the cribriform plate, that the olfactory bulb gets damaged, and it's no different here. I don't have full intact olfactory bulbs. They're just sort of there. So here's our olfactory bulb, and you can see the white matter that will extend posteriorly into here, into the brain. This is our olfactory tract. They typically also always cut the optic nerves very short. So the optic nerves would be coming out of the brain, or actually coming into the brain right here. Okay, so here are optic nerves. Now remember, the optic nerve fibers will cross when they get to the brain. So this center section here is called the optic chiasma. And then the white matter that is going back into the brain, these are our optic tracts. Now remember the difference between an optic nerve and an optic tract. Optic nerves are in the peripheral nervous system. They're outside of the brain. Once they get into the brain, they are now called tracts. Our next cranial nerve is this one, right here. And there should be a matching pair on the other side, right here. So I have two. Good. These are my cranial nerve number 3. This is ocular motor. Okay, so 1, 2. Cranial nerve 3, ocular motor. I have an intact cranial nerve 4 on this side of the brain. It is right here. Does everybody see this? This is very, very thin, very tiny, coming out of the transverse fissure. This is cranial nerve number 4, trochlear. And then cranial nerve number 5 is right here and right here. Now, depending upon how much you got out of it, it might be just a little stub, might be a little white spot on the side of this structure, but this is cranial nerve number 5, trigeminal, here and here. So let's go through the structures on this inferior view from anterior to posterior. Again, we have the olfactory bulbs and tracts. Then we have the optic nerves, optic chiasma, and optic tracts. Just posterior to it, you will see sort of this circle that may or may not look like a hole. Sometimes it looks kind of like a hole right here, so there's a circle here. This is the connection for the pituitary gland. It is the infundibulum. So it is just posterior to the optic chiasma. You have your infundibulum. Posterior to the infundibulum. And sometimes you might have to take some of the connective tissue so that you can see it well. Sort of pull on it. You will see this sort of V-shaped structure right here. This V-shaped structure are the mammillary bodies for the sheep's brain. So these are your mammillary bodies. Surrounding the mammillary bodies, you will see this structure here and this structure here. These are the cerebral peduncles. the ocular motor nerve will leave from the cerebral peduncles. So you'll see the ocular motor nerve attached to the cerebral peduncles. On either side of the cerebral peduncles, you see this structure and this structure. These structures are special lobes that we find in sheep brain, but not so much in the human brain. This lobe is called the piriform lobe. So we have the piriform lobe. The piriform lobe means that it's shaped sort of like the fruit pear. So if we turn our brain upside down, sort of like this, you'll see the bottom of the pear, and then you'll see where the stem should be up at the top. So we have the piriform lobe. It's technically part of the temporal lobe, but we're going to call this our piriform lobe. We then have the pons. So here's our pons. And cranial nerve 5 should be coming out the lateral sides of the pons. Here we have the medulla. And then to the very posterior, we have the spinal cord. We are now going to take our brain and turn it so that we are looking at the cerebellum here. And we are going to pull the cerebellum away from the cerebrum by gently bending the brain stem. We do not want to break the brain stem, but we do want to see back into this part of the brain. We want to be able to see this structure back here. Everybody see that structure? Everybody says it looks like a lumpy butt.
Yep. Yes.
The lumpy butt. Remember, the lumpy butt is the corpora quadrigemina. The corpora quadrigemina has 4 hills. So we have 1, 2, 3, 4 hills. The 2 larger, more superiorly located hills are called the superior colliculi. The smaller, more inferiorly located hills are called the inferior colliculi. You may also see, you might have some connective tissue in here that's superior to the superior colliculi, but superior to the superior colliculi right in the center, and I can't see it very well here in my picture. You may also see the pineal gland in the back here. It is just a tiny little thing in the back here. We now want to do a mid-sagittal cut of our brain, and before we do that, we want to tease apart the 2 hemispheres of the brain by taking your blunt probe and lightly tearing through the connective tissue that's holding the 2 hemispheres together. And you want to get down to the bottom of this where you will see white matter at the bottom of the longitudinal fissure. That white matter structure is the corpus callosum. Now when cutting the two halves apart, you want to use your scalpel. And I like to stand. I think it just gives me better leverage, but if you don't want to stand, that's fine. You want to hold your brain steady, and you want to go into the longitudinal fissure and find the exact midline, if at all possible, and then cut straight down.
The whole.
And you're going to cut through the cerebrum, diencephalon, cerebellum, and brainstem all the way out to the spinal cord. So that you have two separate pieces of brain. Now it is almost impossible to get a dead on center mid-sagittal section. There will be slight differences between the two sides that you see. The first, I'm going to focus on this side of the brain right now. And you can see the first thing I'm going to focus on are the ventricles. The ventricles are the fluid-filled chambers that we have within the brain. The first of our fluid-filled chambers is located right here. This is our lateral ventricle, and there should be one on both sides. So you see the second one on this side. Some of you may not see the hole on both sides because you're going to see this small little membrane, very thin membrane, covering your entrance into the lateral ventricle. This thin membrane is called the septum pellucidum. It is the divider between the two lateral ventricles. Here is our third ventricle, right here. From the third ventricle, you will travel through a fluid passageway here, which is sort of this trench that you see. And depending upon how well you cut it, your trench may or may not be there, but I did a pretty good job. I kind of got in the middle here. So here's my trench. This is my cerebral aqueduct, and it will flow down to my fourth ventricle, which is located right here. So again, we have two lateral ventricles, a third ventricle, a cerebral aqueduct, and a fourth ventricle. Now within the ventricles, especially right here, you see this sort of blood vessel structure here, kind of pinkish gray, located in my third ventricle. And you may also see a little bit of it, if you sort of dig for it a little, in your lateral ventricles. This is the choroid plexus. Remember your choroid plexus, what does it do? It makes cerebral spinal fluid. So you'll find some in all the ventricles, but we happen to see it very well here in my third, and sometimes you'll see it very well in your laterals. Let's see if I can find some here in my laterals. Right here, this would be the choroid plexus in my lateral ventricle. So let's look at the structures you can see on this mid-sagittal section of the brain. So on either side of the lateral ventricle, you'll see white matter. The white matter that you see to the superior of the lateral ventricle is the corpus callosum. The white matter that you see to the inferior of the lateral ventricle is the fornix. Remember, fornix means arch, and it will arch over the top of this circular structure. Superior to the corpus callosum, you will find this gyrus. This is our cingulate gyrus. The circle structure here is the thalamus. Inferior to the thalamus, you see gray matter right here. This gray matter is the hypothalamus. The hypothalamus is bounded by the thalamus to the superior, the optic chiasma to the anterior, the mammillary bodies to the posterior, and then the infundibulum would come out, it's inferior. So again, thalamus, hypothalamus, optic chiasma, mammillary bodies, infundibulum. The last structure of the diencephalon is this structure here. This is the pineal gland. It's very common for the pineal gland to maybe be found on only one side of the brain versus the other because it's a small thing and it tends to want to move around a little bit. So sometimes it really does get cut to one side or the other. I happen to have it on both sides, so here's one half of it. The other half is right here. Posterior to the pineal gland, we have the midbrain. The midbrain has three structures. Here we can see the corpora quadrigemina is here. And remember, the corpora quadrigemina is two structures. We have the superior colliculi and the inferior colliculi. The second structure in the midbrain is the cerebral aqueduct. And the third structure is the cerebral peduncles. We will then see the pons. So the pons is right here. And then the medulla. To the posterior, you will see the spinal cord. We also see the cerebellum. And in the center of the cerebellum, we see white matter. That white matter is called the arbor vitae. The last thing we are going to do is we are going to do a parasagittal section. Again, we're going to do the parasagittal section of the cerebrum. I like to take my scalpel blade and use it as a guide. I will put the dull part of the blade up against my longitudinal fissure, and I really want to cut about the depth of my blade away from the midline. So I'm going to take this and just sort of tilt it upwards, and then I'm going to cut straight down. Until I get two sides. When we have this parasagittal section, we can see that on the very outside of the cerebrum, there is gray matter. This outer gray matter, which follows the grooves of the cerebrum, is called the cerebral cortex. Okay, so this is our outer gray matter, the cerebral cortex. Most of the inside of the cerebrum is white matter. But to the anterior, there is some gray matter in the midst of this white matter. And typically this gray matter has white stripes in it. This gray matter is called the basal nuclei. So here are our basal nuclei. Here you see the inside of the thalamus. So the thalamus would be right in this area here. And then you will see the lateral ventricle, or fluids-filled space here. But in the lateral ventricle you will see this sort of fold in it. This fold is the hippocampus. Remember, hippocampus means seahorse, and I cut mine pretty well so that you can actually see it kind of looks like a seahorse. I have a little nose, I've got a head, then I've got the body and the tail that's curling around the bottom of my thalamus. So this is my hippocampus.